Hello everyone, welcome back to another class on regeneration biology. So we have been learning about different tissue and organ regeneration, and now we are picking different organs from a highly regenerative species, that is zebrafish. So initially we will see a brief outline and then we will go deep in depth on various angles and aspects. Okay, so this class will be about a brief outline about the regeneration in zebrafish. We know zebrafish is a well studied or well used organism whose genetics is well studied. It can reproduce year round, and it has got external fertilization, and it has got transparent embryos, and embryos can be injected with the DNA, RNA, and it can also express proteins. And zebrafish is amenable to large scale genetic screens, forward and reverse genetic screens. And zebrafish shares extensive genomic homology and synteny with that of human. Synteny is the order in which the genes are arranged in a chromosome, and the homology is the sequence similarity. That means the functional similarity is attributed to the sequence similarity. So homology and synteny are hallmarks of deciding whether or not a organism is suitable to study a human physiology or genetics. So zebrafish can regenerate central nervous system neurons following a damage. That makes it a more attractive model organism when regeneration biology is concerned. If you look closely, zebrafish retina structure, it is very much similar to that of a human, and it has got a lens, it has got a cornea, it has a retina, as you can see here, and it also have got an optic nerve which is connected to the brain, and the image processing is very much similar to that of in mammals, except that we have got a flat lens, whereas fish has got a spherical lens. And the cell types are also pretty much same. Rods and cones, which are the photoreceptors, and horizontal amacrine bipolar cells, and also the ganglion cells, whereas the only glial cell type, the predominant glial cell type of the retina is called Muller, as you can see here, yellow in color. And broadly, the layers are classified into outer nuclear layer, where photoreceptors are there, inner nuclear layer, where various neurons and the glia are there, and then comes the ganglion cell layer, where the ganglion cell resides. And there are different ways in which you can create a damage in the eye, and injury model can vary, but the regeneration has to happen in the retina no matter what is the method in which you injure the retina. So we use in our lab and a lot of other labs something called mechanical injury, and that is called a needle poke. We use a 30 gauge needle from the back of the eye, and it will create a hole in the retina, and we usually give one poke per quadrant, and this is called a needle poke method of injury. And it's a focal injury. That means the animal's tissue, the entire eye is not affected. It won't go blind completely. Then the neighboring region, undamaged retinal part, will act as a control region. So it will be a better control than the other eye or another fish's eye. So to study the regeneration, we have to use different model organisms and also different transgenic organisms of the same model. One such transgenic model or transgenic organism we use in zebrafish is tuba1 GFP fish. What is tuba1? Tuba1 is part of cytoskeleton. Whose cytoskeleton? Neuronal cytoskeleton. But we haven't used the full length tuba. We used a fragment of the tuba1 promoter. So what do you understand from this? If the promoter is truncated, its expression will not be as it will happen in the native gene. Say for example, if a gene's promoter is 10,000 bases, and if the entire 10,000 base is there, the gene will be expressed as it should in the native condition. But if the 10,000 base I cut down into 1000 base, I got rid of 9,000 bases. Still it is a promoter, but that promoter comes with lot of limitations. It will not work in every cell type. So that is what you should understand by a truncated promoter. So in the truncated promoter 2/1 GFP, what you see is instead of expressing it in entire neurons, it explains in the central nervous system of embryonic zebrafish, not adult. Embryonic zebrafish, as you can see, brain and the spinal cord, it is there, and also it is expressed in the eye. You can see it is expressed in the eye. But whereas if you see, it is there in the retina also. But in the adult retina, it is not expressed. Adult brain, very little it is expressed. And in the adult spinal cord, no expression. But the interesting thing is, the same adult retina, uninjured retina, it is not expressed. But in injured retina, you can see the expression. See, this is the result of a focal injury. Look at here, surrounding area, there is no GFP expression. Look at this area, there is no GFP expression. Look at this sudden area, this is the place through which the needle has gone through. It is a microscopic image, that is why it is looking broader. Normally the needle is very small, 30 gauge needle. And for zebrafish eye, when you take an image in the microscope, it will look larger area. And this vertical group of cells, they are nothing but proliferating group of cells, as you are seeing here. And they are regenerating cells. And if you see the merge, the green and the blue are merging. So what do you understand from here? In the tuba fish, embryonic central nervous system, including the retina, it expresses. In adult, there is no expression, especially in the retina. After injury, it expresses, and it expresses GFP in the proliferating cell. So this transgenic line is extensively used, and these images are taken in 4 days. Why 4 days so special? Because 4 days the time in a needle poke injury, you can see the maximum proliferation happens in 4 days, roughly around here. And by around 7 days, the proliferation is reduced to a bare minimum. So this is what you should keep in mind. Now, which are these cells? Who made these cells to proliferate? The earlier picture we saw that there is a Muller glia cell. Muller glia cell is the assistant cell of neuron. Just like officer has got a personal secretary or a stenographer as a helper. Like that, the Muller glias are the helpers of neurons. They have some hallmark gene expression which neurons don't have. One such gene is called glutamine synthetase, as you can see here, red color in this picture. And this GFP is the same GFP as you saw here. And when you merge it, you see a lot of yellow color cells. So the GS positive, glutamine synthetase positive cell is having GFP expression, and they are merged together, and you end up getting a strong expression of glutamine synthetase indicative of the Muller glia, and GFP expression indicative that it is now responding to the injury. That is not expressed in every Muller glia. It is expressed in those Muller glia. GFP is expressed in those Muller glia which entered the cell cycle. That is what you should keep in mind looking at this picture. Now, if you can generalize what is happening. This is normal retina. You have nuclear layer, and you have inner nuclear layer, and you have got ganglion cell layer. After injury, what happens? There will be a response. It can be anything. Response can be anything. It can be cytokines, chemokines. It can be any damage, any cellular damage will create some unrest in the environment. This unrest will be sensed by some cells such as Muller glia. Muller glia have got a phagocytic property also. Phagocytosis means engulfing this damaged portion. When it is engulfing this damaged portion, it will sense there is an issue, there is a damage, which make them to reprogram. We have already discussed what is reprogramming, dedifferentiation, etc. Once again, I will tell you, reprogramming means like if you are studying in bachelor's, you are pushed into kindergarten. That is reprogramming. Once reprogramming happens, then they undergo proliferation, and they are called Muller glia derived progenitor cells. Because of this proliferation, they will enhance in number and they migrate into different layers. And different layers, when they migrate, outer nuclear, inner nuclear, ganglion cell layer, they will now give rise to retinal neurons and the Muller glia itself, what is present in the eye. So whatever lost cell type will be restored back, and net result, you will restore back after this migration, you restore back the normalcy. How long it takes? It roughly takes around 2 to 3 weeks time. The whole process will be done with a 30 gauge needle poke. So what you understand from here, because of injury, they are restoring the normalcy in around 3 weeks time involves lot of factors, and it raises lots of questions. What are the genetic factors controlling the Muller glia's dedifferentiation or reprogramming and its proliferation? And it is redifferentiation. It should come back also. Like you are studying in bachelor's, you became a kindergarten, you from that phase now you are coming back into bachelor's. Not the same, but one fellow became medicine, one fellow became engineer, another fellow became advocate, and another fellow became a chartered accountant. This is called redifferentiation. So in other words, what you can see here, the Muller glia is capable of giving rise to Muller glia derived progenitor cells, as you can see here. But for this process, there has to have lots of signaling events such as Wnt signaling, FGF signaling, insulin IGF PA3K pathway, HBEGF MAP kinase pathway, and EGF R pathway. These all contribute, but these are all stimulating the reprogramming event. But some pathways such as delta notch signaling, they are not stimulating, but rather they are restricting. That means restricting basically means just like you are tying your dog to a leash. Like you have a dog, if you unleash it, it will run around. But by tying onto a thread, you are not completely stopping its movement, but you are restricting. So dog can move as long as the length of the leash is there, that much around. If it is a leash length is 1 meter, the dog can move around 1 square meter area. If it is 5 meter, it can go 5 square meter area. Same logic applies. Delta notch signaling is not inhibiting reprogramming, but it restricts the zone in which the reprogramming or redifferentiation can happen. And there are other factors such as INS-M1A, which are inhibitors, whereas ASCL1A, Linton theater, these factors are activators of reprogramming. So there are many transcription factors which will favor the formation of the Muller glia derived progenitor cells, but there are other factors which do not favor that much. That means something like a brake and accelerator in a car. You cannot say I want a car which has got only accelerator, because you can't stop it. You can't do crash every time and stop the car. So brake also equally important as that of the car. So transcriptional activators are important, but when to end the show, when to stop the show, that has to be decided by inhibitors such as INS-M1A. One principal inhibitor is LET7 microRNA. LET7 is a microRNA that is expressed in every differentiated neuron to maintain the homeostasis. What is homeostasis? Neurons do not want to divide. Neurons want to stay in a fixed state of animation. They will do their metabolic work, their physiology, every job will be done, but they do not proliferate. What if there are some genes that are pro-proliferative got turned on? Then the neurons will be compelled to proliferate, or neurons will get into some unrest situation. So you don't want that to happen. Because of that, the LET7 microRNA, it makes sure that these, even if these pro-proliferative genes are turned on, they will never get translated. RNA is not doing any harm. So the LET7s are the guardians. They are the guardians of the maintenance of the differentiated status. So we can say when a differentiated Muller glia, which has got high level of LET7, has to become reprogrammed or de-differentiated into progenitor cells, you want to get rid of LET7. If LET7s are present, LET7 microRNA are present, then the so-called proliferative phase or the next phase will not happen. So we can call LET7 as a principal inhibitor of the glia's migration into progenitor path. So what you can see is there are different way of dealing with, different way of analyzing the regeneration event. So this is from a review, recently published a review. It's a timeline of Muller glia reprogramming and retina regeneration in zebrafish. So you can see there are different phases, and you can also notice that there are different timelines. 1, 2, 3, 4 it's mentioned, but 1 DPI some events happen, 2 DPI some events happen, 4 DPI some events happen, and 7 to 21 days some other events happen. So every process is initiated from a Muller glia reprogramming event. And it's also important to note that every Muller glia, if the retina has got 1000 Muller glia, in that injury spot, I'm not talking about the whole retina, near the injury spot, if there are 1000 Muller glia, every Muller glia will not reprogram. Only selected number, say 100 Muller glia, will reprogram. What stopped other Muller glia from reprogramming? Of course, we can justify why they don't. If a job can be done by one person, we don't want 50% to be recruited. That is understood. But who chose that one person? This is the question you should have in mind. So in the uninjured retina, Muller glia are embedded in a sea of neuron. Retinal injury results in neuronal damage, and this stimulates a partial reprogramming event in the very beginning, in the uninjured retina. So any insult that happens, like I told you earlier, there will be release of some factors which will be sensed by the Muller glia. You can see these abbreviations are there. DPI means days post injury, MG for Muller glia, V for vascular endothelial, VEGF is for vascular endothelial growth factors. And here you can see in different phases, like you mentioned number 1 and number 2, right? Number 1 in MG, that is characterized by ASCL1A and Lin28 expression. Remember, ASCL1A can upregulate Lin28 also. Lin28 can be upregulated by various other pathways, but ASCL1A is one of the members that can upregulate Lin28. Around 1 DPI, 1 day post injury, immune cells, mostly from the microglia, they accumulate near the injury site. Okay, microglia are other wandering type of glial cells that comes into the retina. They accumulate near the injury site and appear to stimulate a second reprogramming event. First, Muller glia will sense these damaged particles and it is vulnerable for reprogramming, but the presence of microglia into the injury spot creates a second reprogramming event. As you can see here, neuronal damage 1, and then comes the 2, second reprogramming event, and that allows for Muller glia division. The second reprogramming event stimulates a division response. That means it has to divide, increase in number. Muller glia undergo as a self-renewing asymmetric cell division. Asymmetric cell division is very important because you want to have a differential distribution of delta notch ligand. Many cells will have both. Like let's say, for example, a rich father is distributing, say if he has got 3 restaurants, or he has got 4 restaurants and 4 movie theaters. So if he is distributing, he will give restaurants to one son and the movie theaters to another son. He will never distribute 2 restaurant and 2 movie theaters to one son and 2 restaurant and 2 movie theaters to other son. Why? Because then there will be competition between them. So the asymmetric division makes sure that there is a differentiality between them, which allows them to have a differential fate. One cell will differentiate, the other will proliferate. So asymmetric division is a necessary. It's not happening because of a mistake. MG undergo a cells renewing asymmetric cell division, producing multipotent progenitor that transiently amplifies before differentiating into one of the major retinal cell types. Because retina has got 6 retinal neurons and 1 glia type. So it can give rise to any of them, but based on the demand, based on the need. So this is what you should keep in mind, that this is a multi-step process. Retinal injury and Muller glia reprogramming that happens, sensing this picture, yellow color cell is a Muller glia, and then it is responding to the microglia present. As you can see here, immune cells, the color code is given: immune cells, damaged neurons, uninjured neurons are gray color, damaged neurons are pink in color, and then Muller glia derived progenitor spindle shaped cells, and the Muller glia undergoes a division, and then the progenitor starts getting amplification, and the final phase 7 to 21 days these progenitors migrate and differentiate into various neuronal cell type. And this two-step process of Muller glia reprogramming in the injured retina is also allowing a controlled response. You don't want a pan-retinal response. So there has to be, it is just like, say you got one phone call from someone, some unknown number, that some trouble is there with the so-and-so person. You will not immediately get into panic. You will call someone more reliable and you ask, I heard so-and-so, is that true? So this is a second level of confirmation. So your two level of reprogramming ensures that you don't want a alarming response, an unwanted alarming response. So in what happens in this picture, there is a two-step has been given. One is Muller glia reprogramming by the damaged neurons. You can see here there is a neuron injury and there is expression of some of the ligands of delta notch signaling, DLL4, DLLB, and there is also this will trigger delta notch signaling, and this will facilitate the Muller glia reprogramming. And you can also see what happens, that some of the cells, that is the VE, vascular endothelial cells, because the newly formed cells also need blood supply. So the endothelium has to assemble so that the blood vessels can form. So the same neuronal injury can create a VEGF expression in the Muller glia, vascular endothelial growth factor, and that can inhibit the expression of DLL4. Unlike the stimulation what you see here, it can inhibit, and which it can favor the production of notch, and that also can favor a Muller glia reprogramming. So you can understand, and also various neuronal signaling comes into picture, like GABA and glutamate, they all come into picture. We will not dwell into those right now. So injured neurons initiate the first Muller glia reprogramming event and involves notch signaling inhibition via the suppression of DLL4 and DLB, which are the ligands of the notch signaling. So notch signaling will be suppressed in neurons, and DLL4 expression will be favored in the VE cells, that is vascular endothelial cells, which is needed for the formation of the blood vessels. Suppression of quiescence promoting factors such as TGF-beta and FGF8, TGF-beta3 and FGF8 by unknown mechanism, and suppression of GABA signaling from HCs, that is you can see here HC cells are mentioned, horizontal cells are mentioned here, and which are present in the inner nuclear layer. These HC cells, they are done via reduced glutamate from the PRs. And the PR basically the what is present in the photoreceptor layer, or the rods and cones which is present in the photoreceptor layer. The second reprogramming event is initiated by the microglia, which is distributed by the immune cells accumulating at the injury site. The mechanism underlying this second event is unknown but may involve TNF signaling. TNF means tumor necrosis factor alpha, TNF-A or TNF-alpha usually, but may involve the TNF signaling, which is because of the presence of the microglia. Note that the TNF-A from the injured and also from the dying neurons has been suggested to stimulate the Muller glia reprogramming. So many cells store these factors when they are present in them in an inactive form. When they are broken or damaged, they will get released immediately outside. So this is how this TNF alpha becomes available to the neighboring cell. So potential roles of notch ligand delta A, C, D induction in the Muller glia derived progenitor cells. So here you can see notch ligands act on the neighboring progenitors to stimulate their proliferation, because delta and notch, they are present, they do a juxtacrine signaling. It is just like two rooms of a hall. Like in many hotels you would have seen rooms are arranged in a hall. So basically if you make a hole in one room, you can enter into the other room. That's how they are arranged. So that is called, so you made a hole in the hotel wall and you are communicating to the neighbor. So this is, you can call this as a juxtacrine signaling. So how does it work is, every cell can have delta, every cell can have notch. So in a given cell, if delta is present and binding onto the notch of the neighboring cell. So the notch expressing cell will proliferate. But on the other hand, the same notch expressing cell may also have delta, and that will be binding onto the notch of the previous cell. Then both will not proliferate. Although both have got delta, both will have notch. So here comes that earlier I mentioned, the asymmetric division. Because of the asymmetric division, there will be a differential distribution. Because if a given cell has got 100 delta and 100 notch, when it divides, it is not split to 50 delta and 50 notch. It will be split such a way that one gets 60 delta and 40 notch, the other gets 60 notch and 40 delta. This way, this asymmetric distribution will have an upper hand. So the more delta expressing cell will not proliferate because it has got less notch, and the other way around, the more notch expressing cell will have less delta. Hence, it will proliferate. The more notch expressing cell will proliferate. So this idea you should keep in mind. And the notch ligands act on the neighboring progenitors by juxtacrine signaling. Notch ligands act on the neighboring progenitors to stimulate their differentiation. So the notch expressing cell will not differentiate. It will proliferate. But the notch expressing cell tell the delta expressing cell to differentiate. Don't proliferate, differentiate into a retinal neuron. And notch ligands act on their parent reprogrammed Muller glia to help return them to the basal state. This also happens. Like you can see in this stage 1, progenitor proliferation, stage 2, progenitor differentiation, and stage 3, return of Muller glia to the basal state. Because many times the reprogrammed Muller glia must return back to its normalcy. So otherwise it will remain in a reprogrammed state, which is useless once the job is done. Like you may have seen, whenever there is a spiritual function going on, you will find a lot of local shops that is coming up. Once the festival is done, local shops all will disappear. What is the point of having those local shops? So these Muller glia must return to their normalcy. And this is a typical in situ injury pattern in the retina. This is a LIN28 gene expression, because earlier I talked about LET7, and the killer of LET7 is LIN28. And you can take a small portion, as you can see in this box, a small portion, and you can see under the microscope. So LIN28 is an inhibitor of LET7 microRNA. And LIN28 is one of the pluripotency factors, like IPS factor, one of the the Yamanaka and Thomson factor. Of those 6 factors, one of them is LIN28, expressed in the embryonic stem cells and cancer cells. LIN28 is found both in the nucleus and cytoplasm and regulates LET7 microRNA levels. LET7 is a very interesting molecule. It has got independent genes, and it can also be embedded in the introns of other genes. Okay, in both ways it is present. And it is produced as a primary LET7, which is processed by DROSHA and DGCR8 to become pre-LET7, preliminary or precursor LET7 microRNA, because this portion is chopped off, and you end up getting, and which is exported to the cytoplasm and acted upon by dicer here. It is acted upon, and you end up getting a LET7 microRNA. And this will help maintaining the differentiated status. But LIN28 can degrade the pri-LET7 bound DROSHA DGCR8, and also it can cause the mature LET7 to degrade by polyuridinylation. So it makes sure when LIN28 is present, it makes sure that there is no LET7 present in the cell. And the downregulation of LET7 is necessary for regeneration. Why? Because many pluripotency factors are induced during retina regeneration. But these LET7 microRNAs have binding site in those RNA, ACL1A, HSPD, LIN28 itself, PAC6, KLA4, c-MYC-A, c-MYC-B, OCT4. So you can see there is an increased concentration of LET7. You see a reduced protein expression that is present. Those who are interested can read this article, what is mentioned here. So this is how absence of LET7 allow the translation. Presence of LET7 prevent the translation of these genes. So we'll study about regeneration biology in the next class more in detail. Thank you.